Lipoprotein lipase is a member of the lipase gene family, which includes pancreatic lipase, hepatic lipase, and endothelial lipase. It is a water-soluble enzyme that hydrolyzes triglycerides in lipoproteins, such as those found in chylomicrons and very low-density lipoproteins, into two free fatty acids and one monoacylglycerol molecule. It is also involved in promoting the cellular uptake of chylomicron remnants, cholesterol-rich lipoproteins, and free fatty acids. LPL requires ApoC-II as a cofactor. LPL is attached to the luminal surface of endothelial cells in capillaries by the protein glycosylphosphatidylinositol HDL-binding protein 1 and by heparin sulfated proteoglycans. It is most widely distributed in adipose, heart, and skeletal muscle tissue, as well as in lactating mammary glands. Synthesis: in brief, LPL is secreted from parenchymal cells as a glycosylated homodimer, after which it is translocated through the extracellular matrix and across endothelial cells to the capillary lumen. After translation, the newly synthesized protein is glycosylated in the endoplasmic reticulum. The glycosylation sites of LPL are ASN43, ASN257, and ASN359. Glucosidases then remove terminal glucose residues. It was once believed that this glucose trimming is responsible for the conformational change needed for LPL to form homodimers and become catalytically active. In the Golgi apparatus, the oligosaccharides are further altered to result in either two complex chains, or two complex and one high mannose chain. In the final protein, carbohydrates account for about 12% of the molecular mass. Homodimerization is required before LPL can be secreted from cells. After secretion, LPL is carried across endothelial cells and presented into the capillary lumen by the protein glycosylphosphatidylinositol anchored high-density lipoprotein binding protein 1. Structure: the crystal structure of LPL has not been discovered. However, there is substantial experimental evidence and structural homology between members of the lipase family to predict the likely structure and functional regions of the enzyme. LPL is composed of two distinct regions, the larger N-terminus domain that contains the lipolytic active site, and the smaller C-terminus domain. These two regions are attached by a peptide linker. The N-terminus domain has an I plus or minus /I squared hydrolase fold, which is a globular structure containing a central I squared sheet surrounded by I plus or minus helices. The C-terminus domain is a I squared sandwich formed by two I squared sheet layers, and resembles an elongated cylinder. Mechanism: the active site of LPL is composed of the conserved SER132, ASP156, and his 241 triad. Other important regions of the N-terminal domain for catalysis include an oxyanion hole, a lid region, as well as a I squared 5 loop. The ApoC-II binding site is currently unknown, but it is predicted that residues on both N and C-terminal domains are necessary for this interaction to occur. The C-terminal domain appears to confer LPL-A unregistered trademark S substrate specificity. It has a higher affinity for large triacylglyceride-rich lipoproteins than cholesterol-rich lipoproteins. The C-terminal domain is also important for binding to LDL-A unregistered trademark S receptors. Both the N and C-terminal domains contain heparin binding sites distal to the lipid binding sites. LPL therefore serves as a bridge between the cell surface and lipoproteins. Importantly, LPL binding to the cell surface or receptors is not dependent on its catalytic activity. The LPL non-covalent homodimer has a head-to-tail arrangement of the monomers. The SER-his triad is contained in a hydrophobic groove that is blocked from solvent by the lid. Upon binding to ApoC-II and lipid in the lipoprotein, the C-terminal domain presents the lipid substrate to the lid region. The lipid interacts with both the lid region and the hydrophobic groove at the active site. This causes the lid to move, providing access to the active site. The I squared 5 loop folds back into the protein core, bringing one of the electrophiles of the oxyanion hole into position for lipolysis. The glycerol backbone of the lipid is then able to enter the active site and is hydrolyzed. 2 molecules of ApoC-II can attach to each LPL dimer. It is estimated that up to 40 LPL dimers may act simultaneously on a single lipoprotein. In regard to kinetics, it is believed that release of product into circulation is the rate-limiting step in the reaction. Function: LPL encodes lipoprotein lipase, which is expressed on endothelial cells in the heart, muscle, and adipose tissue. LPL functions as a homodimer, and as the dual functions of triglyceride hydrolase and ligand bridging factor for receptor-mediated lipoprotein uptake. Through catalysis, VLDL is converted to IDL and then to LDL. Severe mutations that cause LPL deficiency result in type I hyperlipoproteinemia, while less extreme mutations in LPL are linked to many disorders of lipoprotein metabolism. Regulation: LPL is controlled transcriptionally and post-transcriptionally. The circadian clock may be important in the control of LPL mRNA levels in peripheral tissues. LPL enzymes are regulated differently depending on the tissue. For example, insulin is known to activate LPL in adipocytes and its placement in the capillary endothelium. By contrast, insulin has been shown to decrease expression of muscle LPL. Muscle and myocardial LPL is instead activated by glucagon and adrenaline. This helps to explain why during fasting, LPL activity increases in muscle tissue and decreases in adipose tissue, whereas after a meal, the opposite occurs. Consistent with this, dietary macronutrients differentially affect adipose and muscle LPL activity. After 16 days on a high carbohydrate or a high-fat diet, LPL activity increased significantly in both tissues 6 hours after a meal of either composition, but there was a significantly greater rise in adipose tissue LPL in response to the high carbohydrate diet compared to the high-fat diet. There was no difference between the two diets' effects on or insulin sensitivity, or on fasting LPL activity in either tissue. The concentration of LPL displayed on endothelial cell surface cannot be regulated by endothelial cells, as they neither synthesize nor degrade LPL. Instead, this regulation occurs by managing the flux of LPL arriving at the lipolytic site and by regulating the activity of LPL present on the endothelium. A key protein involved in controlling the activity of LPL is ANGPTL4, which serves as a local inhibitor of LPL. Induction of ANGPTL4 accounts for the inhibition of LPL activity in white adipose tissue during fasting. Growing evidence implicates ANGPTL4 in the physiological regulation of LPL activity in a variety of tissues. Pathology: lipoprotein lipase deficiency leads to hypertriglyceridemia. In mice, overexpression of LPL has been shown to affect insulin response and to promote obesity. A high tissue LPL response to a high carbohydrate diet may predispose toward fat gain. One study reported that subjects gained more body fat over the next 4 years if, after following a high carbohydrate diet and partaking of a high carbohydrate meal, they responded with an increase in adipose tissue LPL activity per adipocyte, or a decrease in skeletal muscle LPL activity per gram of tissue. Interactions: lipoprotein lipase has been shown to interact with LRP1. It is also a ligand for I plus or minus 2M, GP330, and VLDL receptors. LPL has been shown to be a ligand for LRP2, albeit at a lower affinity than for other receptors. However, most of the LPL-dependent VLDL degradation can be attributed to the LRP2 pathway. In each case, LPL serves as a bridge between receptor and lipoprotein. While LPL is activated by ApoC-II, it is inhibited by ApoC-III. In other organisms, the LPL gene is highly conserved across vertebrates. Lipoprotein lipase is involved in lipid transport in the placentae of life-bearing lizards. Interactive pathway map: click on genes, proteins and metabolites below to link to respective articles. References. Further reading. External links: Gene Reviews NIHUW entry on familial lipoprotein lipase deficiency, gene therapy for lipoprotein lipase deficiency. Lipoprotein lipase at the U.S. National Library of Medicine. Medical subject headings.